Fourth International Conference on Applied Zoology 2021, organized by the Applied Zoological Society of Pakistan and Quaid-e-Azam University, Islamabad, Pakistan. Assalamu alaikum. I am Mutahira Subhan. I am field scholar, Department of Microbiology, Cohort University of Science and Technology. My research topic is control of disease caused by the Phytophthora capsicii in paper plants using the soil-borne bacillus species isolated from Cohort Khyber Pakhtunkhwa. Contents of my presentation is introduction, rationale, objectives, materials and methods, results, conclusion, references. Introduction of research is Phytophthora blight disease is caused by Phytophthora capsicii, a widespread soil-borne fungal pathogen. It is responsible for the severe damage to paper plants, mainly when the soil contains the excessive moisture after heavy rainfall or irrigation. Paper capsicum annum or capsicum frutescens is in the economically significant and valuable cash crop belonging to the family Solanaceae. Paper crops cultivated over an area of 1,776,000 hectares throughout the world, with the production rate of 7,182,000 tons per annum. Biological control can be a promising and sustainable alternative for in the effective control of Phytophthora blights disease due to their environmentally friendly nature, avoiding the phototoxicity and fungicide resistance. The rationale of my study is the present study is designed to evaluate the efficiency of the soil-borne bacterial isolate against the Phytophthora capsicii responsible for the Phytophthora blight disease in paper crop. This study, imported as the application of the biological control agent, is less costly and cheaper than any other chemical methods. Moreover, biocontrol agents give the protections to the crops throughout the crops period and also suffer to the plants, environments, and in the person who applied them. My research work have three objectives. First one is isolation and identification of Phytophthora capsicii from infected paper plant. And second one is isolation and screening of the soil-borne antagonistic bacteria against P. capsicii. And third one is assessment of in vitro, and in vivo assay to determine the biocontrol potential of the selected bacterial isolates against P. capsicii. Materials and method. First of all, I have collected two types of sample, and for a sample of fungus, and second one is the sample of bacterial species, rhizospheric sample. First sample collection, sample collection for P. capsicii. Sample collection infected plants, parts, for example, leaves, stems, and roots. Sample collected from in the different area of district Cohort, Pakistan. Sample proceeded to the Department of Microbiology, Cohort University of Science and Technology, and isolation of P. capacity through PDA media and SDA media. Identification of P. capacity through special carrot agar media and morphological characteristics. And the isolation and identification of bacteria, antagonistic soil-borne bacteria is a serial dilution and LP media. Identification of bacteria through different biochemical tests and morphological based. In vitro assay, antagonistic assay against P. capsicii, and in vivo assay, the in vivo assay against the P. capsicii responsible for the disease in paper plant for vegetable field and of Cohort. After Result and methodology. The methods and methods and methodology. The result of my present research work is identification of pCAPSicide. The first picture show in the identification through the microscopy, and the second picture show the identification of pCAPSicide through the special carrot agar media. This table shows the morphological identification of pCAPSicide through the microscopy. I have observed the asexual spore sporangia. The range of sporangia length is 44.7 micrometer to 53.1 micrometer, and width is 24 micrometer to 38.4 micrometer. And next, I have observed the shape of sporangia. Shape of sporangia is pear shape. Color of sporangia is colorless, have no color. Papillate pointed at the tip. Spores fall from the colony. Long pedicle stay attached in the base of the spores. This table shows the biochemical test for in the Bacillus subtilis bacteria species. I have isolated 7 different isolates as 1, 2, 3, 4, 5, 6, and 7. I have performed different biochemical tests like gram staining, endospore staining, catalase, oxidase, starch hydrolysis, VP, citric, NECL 6.5. The first two tests on gram staining and endospore stainings show they all of positive and rod shape, and catalase test all positive, but S7 show negative, and oxidase, the first one is show positive, other two negative, and S4, 5 show positive, and last two show negative. And starch hydrolysis, the first one show positive, 3 show negative, and the last 3 show positive. VP test first show negative, second show positive, and 3 to 7 show negative result. And citrate test, first one show S1 show negative, and all of show positive result. And NaCl 6.5 for two different temperature incubation with two different temperature, 37 and 67 5 centigrade. The first 37 centigrade S1 show 37 centigrade and 65 centigrade temperature show negative result, and S2 show positive, both of positive, and S3 37 show negative and 65 show positive result, and S4 both of show negative result, and S5 37 show negative and 65 positive, and S6 show 37 show negative and 65 show positive result, and S7 both of show negative result. The activity of Bacillus species, this is the chart, show the Bacillus activity of Bacillus species. Bacillus subtilis, the Bacillus subtilis show the 90.70% activity, and the Bacillus cereus, the other species of Bacillus, show 90.30%. In vitro and antagonistic assay against the pCAPSicide Bacillus subtilis, the picture A showed the control group and the picture B show the activity group. And the picture Bacillus subtilis show 6 millimeter zones. And this one is the in vitro antagonistic assay of pCAPSicide Bacillus cereus. The A picture show control group and the second one show activity, and Bacillus cereus show 11 millimeter zone. This is the flow chart of in vivo assay. First of all, broth formations, a nutrient broth for broth Bacillus subtilis and the PDB growth for P. capsicii. The incubation period of B. septilis for 24 hours and P. capsicii for 5 days incubation. After incubation serial dilutions, prepare 1 mole of diluted P. capsicii put into 50 ml distilled water and 1 mole of diluted bacillus species put into 50 ml distilled water. After serial dilutions, both diluted solution apply on the healthy plant, spray on the plants, and after 15 days, the Bacillus subtilis show the activity against the P. capsicii. This is the in vivo assay. After the application of fungus Bacillus species, this is in this picture show the in vivo assay. The conclusion of my research, this study concluded that Bacillus subtilis show the activity against the P. capsicii, which also promote the plant growth. This is the references of my research. Thank you for your attention.